at the North Carolina Museum of Natural Sciences in beautiful downtown Raleigh. It's Science Tonight. Now here's your host, Chris Smith.
Hey, good evening, everybody. Welcome to Science Tonight. I am your host, Chris Smith. It's great to be with you this Thursday evening. It's been a fabulous day hanging out, just enjoying all of the great spring weather that we're now having here in the central part of North Carolina. It's great to have days where the sun stays out a little bit longer and you can enjoy the wildlife and the plant life and just getting to spend some time in nature. I hope that everybody is enjoying their day, enjoying their week so far. It's good to be with you. If this is your first time tuning into Science Tonight, let me know. Drop a note in the chat over on YouTube or in the comments on Facebook. Say hello, hang out with us because we've got a great guest tonight. We're going to learn a lot. We're going to talk about some really cool topics, I think. Before I jump into the program for tonight, though, we'll do a little bit of housekeeping. I want to tell you about a cool event that's coming up just next week. Next Monday through Saturday, so the 19th through the 24th, I think is the dates, is the Triangle SciTech Expo. We will be hearing from experts across science, technology, engineering, mathematics, and more all week long as part of the SciTech Expo. You can get information about this big major event that the museum is throwing. It's all virtual at naturalsciences.org. You can register for all of the programs that you think you'd like to participate in, and you'll get reminder emails when it's time to click the link and come and join us. You can also find almost all of the programs that'll be live right here at the museum's YouTube channel. So another great thing to do is just subscribe right here to the YouTube channel and then click the little bell icon to get notified. And every time we're bringing you great content out of the museum, you'll get the notification right to your device and you can come and join us. So there you go. Triangle SciTech Expo, naturalsciences.org. Now, let's get to the Science Tonight part of the show. Tonight, we're going to be talking about wildlife and conservation genetics. My guest is Dr. Elizabeth Kierepka. Dr. Kierepka is fairly new at the North Carolina Museum of Natural Sciences as a research biologist and, I hope I get it right, associate professor at North Carolina State University, where she's joined assistant professor. I always get the two mixed up. But Liz, thanks for being here.
It was great to be here. I'm happy to interact with everybody tonight and get to know sort of the folks that are here in Raleigh and across the country and world.
So remind me, when did you actually start at the museum?
I started in September 2020. Yes, it was. So I've been here since pretty much the middle of the pandemic and throughout the later part of 2020 and into 2021. So I haven't known anything different so far.
Well, I think I recognize those pale gray walls behind you. How much time have you been able to spend in the lab at the museum?
Not much. And that's probably due to the fact that because I do genetic work and a lot of the materials that are needed for our vaccines and testing for COVID are also needed for what I do. So it's just, there are delays with things. So it's easier to get sort of these other types of activities done before I start doing actual lab work, just because of the time sensitivity of a lot of things.
Oh, wow. That's fascinating. Very interesting. We might have to dive into that a little bit more when we come around. Folks who tune in to Science Tonight on the regular know that I like to start the show off, though, with a laugh and having a little bit of fun. And so all of the guests play trivia. Let's go ahead and bring up the trivia game onto the screen. I've got a special game for you. Since you are a wildlife geneticist, I thought we would talk about some very famous genes in the human population. Celebrity genes.
All right. Oh, okay. So not in DNA. Got it.
Not in DNA at all. No, we're going to turn it to some famous performers. I'm going to play 3 song clips for you. We'll listen to each
Okay.
tune, and then you tell me which gene you think performed that piece.
All
And
right.
of course, I'm calling this game Name That Gene. And everybody in the chat box can clap and say how much fun. So let's take a look at the first one and give it a listen.
Uh-oh.
Advance one more time to play the audio.
Cannot hear.
Yep. Okay. Well, Liz, is it Gene Chandler, Gene Autry, or Gene Campbell?
I did
You
not
have
hear
no
the
idea.
audio.
How would you know?
But not a clue.
I didn't
so
either.
I can.
So let's see if you can just
I
guess it.
can-
Hold on. All right, here we go. I'm improvising.
Okay, I was like, I do know the song, and it's well. I think that's kind of cheating if I say the right answer now, isn't it? It's A.
You know what? It's A. You're right. It's Gene Chandler. Okay.
Perfect.
That went over great. Let's see if we can get the next one.
Perfect. All right.
Oh,
Do
no.
not have audio. But I know who 2 of those people are, so I have a better chance at this one.
Okay, don't worry.
That would be B.
That would be, let's see the correct answer.
Gene Simmons. Gene Simmons.
That is Gene Simmons. Absolutely right. Co-writer of the song Rock and Roll All Night
Yep.
from the band Kiss. Okay,
I have a feeling that was
I'm calling it
was the easy one.
That you think that was the easy one? Okay.
Yeah.
You've got one more. Let's take a look. Now this one is going to take me a little bit longer to find because it was the harder one.
Yep.
Let's see here. So for this one, your options are going to be Gene Krupa, Gene Roddenberry, or Gene Wilder.
All right.
There is no life I know to compare
That's
with your
C, Gene Wilder.
imagination. Living there, you'll be.
Answer C, Gene Wilder. Let's take a look at the correct answer.
My favorite movies as a
Boom.
kid.
Oh, Charlie and the Chocolate Factory. No, Willy Wonka and the Chocolate Factory.
Yep.
Would you say that you were inspired by Willy Wonka to become a wildlife geneticist?
I'm not sure how that would work. I definitely did love the movie, but if anything, it made me apprehensive about going through tunnels with boats.
Ooh, yes, definitely. Yeah, for sure. All right. Well, hey, we may do with that. We may do with that game. And you still got 2 out of 3. And I think you probably would have gotten the first one right, too. So everybody, round of applause for Dr. Kierepka. That puts you in the Science Night Hall of Fame. Well done.
Mm-hmm. All right.
So you're an expert in wildlife genetics. Let's talk now, instead of about celebrity genes, let's talk about the genes that you're most interested in, the ones in wildlife and wildlife populations. Give us a taste of what your job is and what it is you do.
Sure. So what I basically use genetics to answer questions about wildlife. There can be lots of different questions. It can be anywhere from looking at species within these areas where we're trying to figure out how many species are present. So we're looking at these big scale type of continental questions, all the way down to how different are these individuals, and can we tell the difference between individuals and estimate how many there are. So anything in between there is sort of what I use. But essentially what I do is I use a piece of a wildlife individual, a whole group of them. I am able to extract their DNA, amplify their DNA, and then look at the differences between them. And based on where in their genetic code I'm looking at, I can tell the difference between species, populations, individuals, and even looking at all of these different scales, we can look at these both evolutionary processes like gene flow or just the movement of genes in the environment, all the way up to what has caused them to become different species or become conservation concerned species, like if they're vulnerable to things like inbreeding, where they're breeding with their relatives and having poor traits because of that. So that's sort of a general overview, but there are a lot of different questions that I can answer just with this general framework.
Okay, so you're not so much interested in, say, a particular species and all your effort goes into that. You've got this tool of being able to analyze DNA and you just apply it to as many different questions as you can.
Yep, I have pretty broad interests. Generally falls within mammals, but I have done a little bit with birds and snakes as well. But so I think I'm up to 12 species I've worked on, and I'm adding more all the time because each question you like to ask has a certain set of species that really fit well with it. So it just gives me an opportunity to look at many different aspects of wildlife, which is really exciting for me.
So does that mean you go out into the field and you're, I don't know, plucking hair off of all the different mammals in order to study them?
So it certainly depends on the species. So when I worked on, during my PhD, I worked on American badgers. And yes, I was the one mostly going out into the field and setting up these hair collectors on their burrows, which the badger would then pass under it and it would pluck their hair off. Other times I rely on things like hunted individuals for like deer or individuals that are critically endangered, like the gorillas I've worked on, or endangered now. They rely on fecal DNA. So you can get DNA from pretty much any material that that an individual leaves behind. So for individuals that are really hard to find, fecal DNA works really well. And that just means we're collecting their poop instead of the individual. So it really depends on the species. Some of the snakes I've worked on, we actually handled the snakes. But in general, I tend to be more in the lab than the field. I usually have wonderful collaborators throughout different agencies and actually citizen scientists as well that help me identify individuals and collect them. So basically depends on the species. That'll probably be a common saying you're going to see throughout this, is that really depends on the different species you're working on.
Okay, very, very interesting. So I don't know, tell me a little bit about some of the species you've worked on then.
Sure. I would say that the one that I worked the most on was the American badger, because I worked on both for my master's and PhD. If you're unfamiliar with them, they are a fierce little guy in the weasel family. They weigh about 30 pounds max, but they are known for their personality. They are one of my favorite species, but there's very little known about them because they are so hard to find. They're nocturnal, they're underground. And so much of that project was understanding their basic biology and understanding what's important for them. There's other species like I worked with mountain gorillas, which were a very well-studied system in Uganda because they are endangered and they are an important tourist opportunity for people to actually go up and interact with these gorillas for an ecotourism. So it's a really interesting system, and that relied mostly on fecal DNA. I've worked with Allegheny wood rats, which are a local species here in the Appalachians, which is a rocky outcrop specialist rodent that is experiencing habitat loss and disease factors. So we really wanted to understand what affects their movement as well as their population sizes. I've worked with also invasive species, which are ones that are overly abundant, things like wild pigs in the southeast, brown tree snakes in Guam. And those are really aimed at understanding the impact these species have on their native environments. And then I've worked with black bears and many different species, coyotes, all over the map. Basically, most of those works have been either estimating population size or looking for areas that are important for their movement. So for bears, for example, how they move in the environment according to human development and things like that. Great.
Okay, well, let's break those two things down then, because I'm curious how you estimate population size from DNA samples. Those two things don't connect in my mind.
Right. So basically what you're doing is you're collecting, in the case of gorillas, fecal samples. So each of those fecal samples was deposited by an individual, but you're going to repeatedly sample these areas, and you're going to sample poop over and over and over. You need to know if those fecal samples belong to the same individuals or different. And so when you have what's called recaptures, where you sampled this fecal pile in one area on this day, and then 3 days later you find them in a different area, you can do that over and over and use a statistical analysis to estimate that population size based on how often you recaptured those individuals and how often you got individuals only once. And so there's a whole host of statistical techniques that can do that. But really what you're after is getting these recaptured individuals based on their fecal samples. So you have to sample enough to get enough individuals in a population sampled over and over, so you can sort of have that upper limit. Because if you just keep sampling unique individuals, that means your population size, you're not sampling individuals repeatedly, so you probably have a very large population size. That's sort of how it's
So in the photo that we were just looking at with the two researchers down, it looked like they were collecting. I'm guessing that was a fecal collection.
Yes.
And I do understand why you would want to know population sizes when it comes to protecting and doing conservation management for threatened and endangered species. But there's also surveyors out who were, I would imagine, out like looking for and counting, say, gorillas. Like they walk through the forest and they say there's 5 gorillas over there. So we know we've got 5 gorillas. Is the DNA like a backup tool? Is it better than the head count?
So in this population, there are 2 different types of gorillas. There are gorillas that are used to humans because they've been habituated. So those are called monitored gorillas. So they won't run away. But in this environment, the foliage is so thick that there's a good half of the population that do not tolerate humans. You will never see them. And so they run away. And so you can get pretty good numbers based on observations of the monitored groups. But what fecal DNA is really important for is looking at those unmonitored groups, because that's over half the population, or approximately half. It really depends on, because they're in various stages of habituation sometimes. And so sometimes they'll run away, sometimes they won't. And then it can just depend on which group happens to be where at that time. So fecal DNA and those observations are both really important because, one, I don't know who those groups are based on just their DNA. With these field crews that go out, and they are some of the best field crews in the world. They know where these groups are located. They can actually tell me if it's likely a male or a female, if it's an infant, based on just the size of their fecal samples. And they can give me a really good idea of what group that is based on what they saw, but also the location. So they're really important to work together with populations like this.
Did you actually get to work with these gorilla populations directly, or were you just getting like coolers full of fecal samples?
I got coolers full of fecal samples. So in this area of the world, it is quite expensive to go over there, but also there were a lot of, there happened to be an Ebola outbreak that was going on nearby and things like that. So it just made more sense for the field crews out there, because they are much better equipped to be able to go out there. And I would probably have been dead weight going out in the rainforest and going up, because this is a mountainous rainforest. And so really it would be me being trained by them. And so as much as I definitely plan to go and do that eventually, in terms of field sampling, it's much better to leave it to the experts in the area. And all of those people are live there and know those forests much better than I ever could.
Makes sense. Makes sense, I guess. Well, so what did you learn then? You process the DNA, you get the results back. Mm-hmm. What did you learn about these gorilla populations?
And so the really important thing of what they're doing with these gorillas is, because there's only one population in Uganda left, and then there's another one in Virunga Massif Volcanoes National Park, and that's it. That's all that's left. So they're really important to estimate population sizes in order to, because they do this through time, they want to see if their conservation efforts are leading to more gorillas or less gorillas. And so really the important finding from this study is we found more gorillas this year than we found 5 years ago. So we have a really good evidence that conservation is absolutely working in these areas. And so that was the main impetus of this project, but also to really hone in on these different interactions that we can have with gorillas, how many groups are within the park, so we can actually identify family groups and how many of those have changed, how many solitary individuals there are. And again, all of this is really important for adapting to these different conservation efforts, and to really be able to conserve, especially these unmonitored groups where we don't really know those things because they're so shy compared to people. And so that was really the important findings from this study. But there's also anthropological data available and many different studies going on within this group. It's a very, very large collaborative project from Germany, within Africa. All of these are really important to conserve this species. Subspecies.
Well, I mean, that's great news to hear.
Mm-hmm.
That such an iconic threatened species, mm-hmm. that conservation efforts are really working, and that there are mm-hmm. huge teams of people dedicated to making sure that they have a secure future. That's great news.
They're the only great ape that actually is increased.
The only great ape that's actually increased in populations.
Yep. Mm-hmm. Yep.
Besides us.
Yep. Yes. we're definitely not on a downward trend. Yeah.
We're not on a downward trend. Well, I did not actually know that about gorilla populations. That is happy news. Mm-hmm.
Yeah.
If everybody remembers one thing, remember gorillas are on the up.
Yeah, this one subspecies, the rest of them are unfortunately definitely on a downward trend. There's two species of gorillas, and then mountain gorillas are a subspecies of the eastern gorilla. So mountain gorillas are the only ones that are doing their populations are increasing. The rest of them are either stable or decreasing.
Got you. Now, I have to admit, when I think about conservation, like big C conservation, mm-hmm. the real big flashy projects, flashy species, right. Of course, gorillas are at the top of the list. But you mentioned badgers at the top of the show early on. And so I'm really curious about this badger research and how that plays into conservation and your work.
Right. So badgers, where I did my PhD, was at the University of Wisconsin, Milwaukee. So Wisconsin loves badgers. It's their iconic species, and they were protected, but they really didn't know much about them. They're actually protected as a species with information needs. And what that means is we don't know enough about them to make any sort of management decision until we get biological data about them. The reason why they're so difficult is because they're so elusive. From what we learned, they maintain these really large home ranges, basically the area they use. They dig these burrows, but they tend to move them every day. So you'll see 30, 40 burrows, and it'll look like 30, 40 badgers did that. And it's actually probably just one. And so they are digging machines. And so what the impetus of my whole PhD was essentially to fill in these gaps about where they occur, how many are there. And that's much more difficult when you're dealing with a species like this. Gorillas are very nice about pooping in areas that you can access. So, but also what's controlling their movement, what's the impact of human development, particularly agriculture, because badgers are thought to be grassland specialists, and grasslands are often converted to agriculture. So really they wanted to know, is agriculture having a negative impact on badgers? And it turns out it's not. Badgers are actually doing very well in Wisconsin. We found really high genetic diversity, which is indicative of a very large population size. So they're doing very well. Their movement, they're experiencing a lot of movement. I found, so I had a citizen science project going on, collecting camera trap data, as well as people reporting badger sightings. And I got badger sightings from almost every county within Wisconsin. Certainly there were areas that were much bigger hotspots for badgers, but they pretty much were found in every single county, even if there weren't really strong tracts of grassland. So it does seem that they were doing very well, which is great, because we didn't know that, and they were thought that they might be on a downward trend because of this conversion to agriculture. It just seems that badgers alter their behavior, or they move more to deal with this habitat type, but they will travel through agriculture, and they don't avoid it. And so, and they can hunt in them as well. They'll just build their burrows on the edge of that habitat, because there are things like mice and woodchucks and 13-line ground squirrels, which they will absolutely partake of. Really, it seems with them, as long as there's a sufficient food supply, and they like to eat burrowing rodents. So mice, woodchucks, or groundhogs, whichever you prefer, all of those are really important for badgers, and those things are pretty tolerant of humans. And so it seems badgers are as well. So in some of these pictures, you see they're in gardens and cherry orchards and things like that. And these pictures were really important for understanding, where these badgers are occurring, as well as collecting genetic samples from them to look at things like that movement, as well as the genetic diversity.
Very impressive.
It was an awesome. They're an awesome species. They're a little difficult, but they're fun.
I know. I think one of the pictures was like a row of badgers headed somewhere. They looked like tiny badgers. They didn't look like adult badgers. So,
So I lovingly call that picture Baby Parade, because that's mom in the back, and then her 3 cubs, basically playing outside the burrow. They gave me hundreds of pictures of this family that was just hanging out in this cherry orchard. And so you can usually tell when they're a juvenile when they don't have that flattened appearance yet. So mom is that one in the back. And then each of the babies kind of still look a little round, and then they'll flatten out as they get older, again, to be able to dig much more easily. But yeah, this is easily my favorite picture that I've gotten of badgers, because it's in a cherry orchard, but there were a really healthy population of chipmunks there. And so that's what they were eating. And so this was right on the coast of Lake Michigan. So it was beautiful area, and they were actually pretty tolerant of humans, which a lot of them, especially mothers, don't tend to be very tolerant. She seemed to be quite tolerant to have babies out in the daytime, and then she knew where the camera was. So probably my favorite picture of all time.
I had to bring it up because that's a really interesting story too. So people and badgers are getting along okay. Like I'm thinking if I'm
In
the
Wisconsin,
orchard owner.
for sure.
Okay, in Wisconsin.
Yeah. I think
what do we know
is
about badgers elsewhere then?
So another part of my PhD was looking at them nationwide. So they have a very large geographic distribution, basically Michigan and Ohio, all the way out to California, down into Mexico, and a little bit up into Canada. So they have a very, very large distribution. And out west, we didn't know too much about them outside of these smaller sites where they had a good population of badgers. And so really the question was, are there anything that stops a badger's movement to create what's called subspecies, usually, or unique populations where they're genetically different from each other? And it turns out that they're just basically one big happy population. They have massive amounts of genetic diversity, lots of movement. Habitat really didn't have an impact. The only thing that we detected as a barrier to movement was the Great Lakes and a little bit of the Mississippi River. And these are gigantic barriers, and badgers are not the best swimmers. So that made sense. But yeah, out west, it's basically one big population. And so it seems that they're doing very well. The few places they're not doing well tend to be their range edges. So in Canada, particularly in British Columbia and Ontario, they are protected and critically endangered in some of these areas because there's just not a lot of habitat, and they're situated right next to very large roads where when they disperse, they get hit on roads. So it really depends on where in their range you are. But for the most part in the United States, they're doing very well. Mexico, we weren't able to get a lot of samples from because it's really tough because they are protected down there as well. And they live in areas that are more remote and in deserts. And so in these areas, it's just harder to access. So we're still trying to figure out what's going on with badgers in Mexico. But besides those few endangered populations, they are doing very well, it seems, at least genetically. Because they are often considered a pest species, particularly out in more ranching areas, because their holes are hazards to cattle and things like that, because they're large burrows and cattle can sometimes step in them. So, but it does seem, despite that, they seem to be doing quite well.
Folks, if you're just tuning in or just tuned in recently, tonight on the show, we're chatting with wildlife geneticist Dr. Liz Kierepka, who works at the North Carolina Museum of Natural Sciences and North Carolina State University. So, Liz, what makes wildlife genetics for conservation challenging? What's the hardest part?
I would say, well, I mean, there's several parts that can be interesting, I guess I would say, is when I'm dealing with species like badgers, where they have these massive geographic ranges, you have to get samples. And sometimes that requires hundreds of people and coordinating all those people and trying to get them to coordinate with each other. And so it's really rewarding to work with that many people, but it can also be really daunting, especially when you're trying to get tissues in the mail, for example, where sometimes they can rot. So getting all of that coordinated. But I would say probably the greatest challenge and also the most exciting thing about wildlife genetics is it's become wildlife genomics. And genomics in the last 10 years, we've moved from just looking at little pieces of DNA to really trying to get these much larger data sets to look at multiple processes, look at different areas that might be important for actual things that do functions, things like immune response, looking at body size differences, diet differences, things of that nature. And that is a really exciting thing, but it has grown so fast. And so keeping up with those methods sometimes can get very daunting because it has required a lot of newer techniques that involve very complex computer programming, as well as statistical analysis that sometimes it just takes a really long time sometimes, because instead of getting, a simple Excel sheet, now you're getting terabytes of data. And so just being able to handle that level of data makes it really exciting, but also can be challenging. And so I would say just the expansive growth that has occurred, particularly in the last 15 years with genomics, is the most exciting part, but also the most challenging for me.
I'll remind everybody who's watching too, jump in the chat box and in the comments and drop your thoughts and questions about tonight's topic, conservation, genetics, into the chat, so that here in just a few minutes, we'll flip over and start to take everybody's questions and hopefully have a good dialogue going. So jump in the chat, everybody. But Liz, I'm also curious, what has been, the most surprising thing to learn? You get the data and you go, oh my gosh.
I think that probably would go to badgers again. I was flabbergasted that they just don't seem to care. I know that there's a very popular meme about how badgers don't care, and it's true. I actually had trouble getting people to believe me sometimes. That I was like, no, I know you would think that, a 30-pound animal gene flow wise would look very different from an animal from Wisconsin, would look very different from an animal in Colorado. And genetically, they looked very similar. And it was just amazing how at these huge continental scales, you can have very little effect of habitat, humans, and especially considering they're an elusive species. They're really hard to track because they're underground. So you can't put a traditional collar on them that you do with a lot of other wildlife. So it was just a really amazing find and made me respect them quite a lot more. And just being able to the complexity of things, because sometimes answers aren't super simple, and just how that can surprise you that all of these different factors can go into what you see genetically. And because it's not just movement, it's not just population size, it's all of those things interacting with each other across time. And so they can have these massive changes that you expect to find something and you'll find something completely different. And so badgers were probably the best example of that for me, just because, again, I expected to have something. And it was a big ball of genetic diversity. And that was, exciting because they're doing really well, but I was expecting them to at least be keying in on grasslands because that's where they tend to be found. And so that's probably the biggest surprise I've had, but I don't think I've ever gotten an expected result. There's always a surprise somewhere, just because there's so many different factors that can impact genetics. So I guess that would be the biggest one that I can think of, but I'm sure every project has something where I was like, really? Okay.
So this is the road we're going to go down now. Well, okay, so I'm thinking about something you said earlier, talking about using the genetics, and I guess now genomics too, to look at the evolutionary histories of some of these animals. And so if you see that all of the badgers in America are pretty similar, what does that tell you about their shared history across very different landscapes?
So what that means is, one, they're pretty adaptable. So one of the most major influences on North America and any temperate region really are the ice ages. So these occurred hundreds of thousands of years ago, all the way up into about a little bit under 20,000 years ago. And so these have been identified as major influences on thousands of species. And so what typically happened is many of the northern areas were covered with glaciers, and then species moved down into these suitable habitats, and then they were separated from each other for thousands of years. And so genetics often picks that up. And so that is what we often can call something like an evolutionary lineage or a subspecies. It depends on how different they are. But what I expected in badgers, at least, is for them, because they have this broad range, species with broad geographic ranges tend to occupy at least two glacial, what we call glacial refugia, where again, they're escaping those glaciers and they're sitting in suitable habitat. Badgers, because they're grassland specialists, we knew the Great Plains would be a big part of that because it's a large grassland. And so what really was amazing about badgers is it does seem that they were able to just persist below those glacial areas all throughout North America. So there's multiple areas where fossils have been found during what we call the Pleistocene, which is during the ice ages. And so it doesn't seem that they were restricted at all. They just were broadly found in these places. And then when glaciers receded, they were able to move back into those areas. And so that's why you can kind of have that really broad geographic range across these different landscapes, but just have really weak genetic differentiation because they were all in genetic communication throughout history. And so that's what's really interesting about these species, because to compare that with another species I'm working on, gray fox, where, you know, they're broadly found in across North America as well, but we have really, really strong, almost species level differentiation between eastern foxes and western foxes, and that was due to the ice ages. And so it's really hard to predict sometimes how the ice ages can impact these species, especially these perceived generalists that don't have a really strong association with any sort of habitat type. And so badgers like treeless areas, but they can live in pretty much deserts, sagebrush, grasslands, you name anything that doesn't have a ton of trees where they can burrow, they're going to be there.
Wow. Fascinating stuff. Absolutely fascinating. So
I think so.
What critters are, yeah, right. What critters are you looking into now that you're in North Carolina?
So I've sort of done a pivot, and I'm gotten really interested in urban adaptation, particularly with probably the species everybody thinks of when they think of cities, raccoons. So they're affectionately called trash pandas for a reason. They do really, really well in urban habitats. And so I've become really interested in how they adapt to human environments, because again, they do incredibly well. But they eat very different diets. They are exposed to probably different diseases. They are, they're probably altering their activity patterns. They're occurring at massive densities in some cases. And so that really provides a excellent template for natural selection to act, because natural selection is going to act on genes that confer a benefit. And so in these populations, have they adapted to these environments, or are they just able to switch to use what's already there that would be found in a rural habitat and just be able to use it and it doesn't affect them much? Or are they actually genomically adapting to these areas? And so that's really been one of the major things I'm interested in North Carolina. I'm continuing my work with gray fox because we just sort of identified a contact zone in sort of where the Great Plains and forests meet in Texas, Oklahoma area. So I'm interested in really diving deeper into that, because we've only just identified that we have these two really strong lineages, and there's a sort of this contact zone, but they don't, at least from our evidence right now, they don't tend to breed very often. And so I'm curious about why that is. Is there some sort of selection against that? So are Eastern individuals only like Eastern individuals, or in Western individuals only like Western individuals? And so they just don't breed with each other, or are there other mechanisms? And so that's been a really big interest of mine. And then also working with wildlife individuals locally, as well as I'm really interested in starting up some citizen science projects, as well as getting involved with outreach with our with Raleigh and our local community. And because I haven't been here this long, I haven't developed specific projects, but that's a really big interest of mine as well, is to sort of bring genetics into sort of a citizen science, or getting undergrads and high school students involved in understanding how doing some lab work, and if they're interested in these ecological or evolutionary questions of local species here. So that's really, I'm of course interested in a lot of species, so I'm very excited to start collaborations and things here as well. So that's sort of the species that I'm working on so far.
It looks like we're going to have lots of opportunities to have you back on the show to talk about all kinds of different species and all of these different projects. This is great. Everybody, let's turn it over to the audience and let's see what all of you folks are thinking. There's still plenty of time left in the show, so if you've got questions or thoughts, drop them into the chat or into the comments, because I'm going to turn to those right now. And it looks like we've got lots of questions here. So Liz, I hope you're ready for some good questions.
Oh, of course, always.
The first one comes from Caitlin. Wants to know what got you interested in using genetics to answer these questions.
I would say I was a person that really liked animals, and I really got interested in how animals move during my undergrad career at Michigan State. And so I really liked doing that, but I also liked being in the lab as well. So my first job, I was actually in a laboratory. And so I really liked combining those two things. And so I found out in one of my classes that you can actually use genetics to do that. And so that sort of really got me interested in these sort of looking at genetics to infer movement. And I was lucky enough to work with Dr. Kay Holkamp at Michigan State and work with one of her graduate students looking at how hyenas moved in relation to cattle. So I also got really interested in how humans impact that movement. And so that's really what started me on my journey through my graduate school of trying to figure out what exactly I wanted to do. And so one of the biggest areas I've worked in is landscape genetics. And that happened to be a new field that was coming out right when I was starting into my master's degree. And so that sort of got my interest. And what landscape genetics does is it combines landscape ecology, which is essentially looking at landscape pattern. So quantifying, how much urban habitat and things like that, and genetics to look at movement and connectivity between populations of species. And so that's really what started me on. It was this interest in landscape genetics. And it just kind of blossomed from there because, again, there's so many different questions you can answer with genetics.
On that note, Caitlin had a good follow-up. What advice do you give students who are interested in genetics?
So I would say that if you can get into the lab as soon as you can, so if you're an undergrad or a high school student, volunteer opportunities, literally email somebody that you think does really interesting work. Most of us are really excited to get people into the lab because lab work is a huge part of genetics. And the more experience you get, the better. Also, because genomics is becoming so much about computer programming as well, if you do have an interest in that, absolutely pursue that because it's going to be such an important part of your career. But if you're younger than that, I would say, there are different ways that you can get experience in labs. There are commercial labs that you can volunteer or get a job in. It's really about just getting exposed as early as you can, because one of the biggest things is because there's so many things genetics can do is what is your interest? And so if you work with somebody either at a university level or at a private level, even in there are different nonprofits that have geneticists, places like Panthera that do big cats, for example, have geneticists. And so really connecting with people in the field, I think, is really important. Absolutely don't be afraid to just cold email somebody. We all welcome people who are really excited because we love doing what we do. And really, one of the biggest part about being a scientist is connecting with the next generation. And so that would be my biggest advice, because it is sometimes really daunting or intimidating, and really don't be intimidated. We always welcome, and myself included, welcome people who are excited about what we do and want to learn. So that would be my best advice. But again, be ready for computers, because they will become a big part, and they're always going to be a huge part of genomics going forward.
Excellent advice. All right, more questions here for you. Troy asks, what is Dr. Kierepka sampled in the Appalachian Mountains, and what was surprising in that area? I think you mentioned the Allegheny wood rat. Is that right?
Yeah. Yeah, so the Allegheny wood rats were sampled by somebody else. I was given samples for that too, but I've also done work with black bears in the Appalachians as well in South Carolina. And so the most interesting part is really, interesting part is how amazing that environment is for how biodiverse it is and how many species and how much we don't know about these things. Because I expected with Allegheny wood rats, for example, that they were going to be really keying in on very certain features within the environment. And I found out that elevation really covered all of that. They really are these rocky outcrop specialists. And so I was so used to doing species where when they, like a badger, when they say it's a specialist, I was like, oh, okay, yes, they are. But in the Appalachians, a lot of species are the greatest example of specialists you can get. They are super associated with their particular habitats. And so that was really amazing to me to see that. And then to compare that with black bears, even black bears where, I sampled from there's a coastal population as well as a Appalachian population. I thought, they're exchanging genes with each other all the time. And I found they actually aren't. They were completely genetically isolated from each other. And so it really just harkens back to how pristine that habitat is compared to, pretty much any other area that I had worked in, especially in the Southeast. I was used to working with things that are much more okay with these more developed habitats, and how these species just really depend on that habitat. And it's just beautiful habitat up there.
Absolutely agree with that. Okay. Let's get into, there's some, what I think are probably really tough questions. Well, tough, good, in-depth questions.
All right.
Glenn is asking, do the rates of genetic mutation vary from species to species, or are the rates similar among different species?
So it depends on what type of region you're talking about. So they tend to vary across species. That is a active area of study. So for example, if you're a species that is small, has a very short generation time, so that means they can replace themselves very quickly. Their mutation rates are expected to be different than something that's larger. Again, there's tons of variation in there. But again, it really depends on mutation rates tend to depend on how important that gene region or area is. So there are some markers that I use that are called microsatellites, which are completely neutral. They don't serve any purpose. They're basically these just tandem repeats of those nucleotides that make up DNA. So there are these little, what you often see is A's, T's, C's, and G's. So it's just these repeated segments, and they mutate very quickly because they're not important. And because they're these repeats, it's very easy for the enzymes that are replicating DNA to slip. So you can get these really fast mutations that are really useful for telling the difference between individuals. Then there are things that are really important. So a mutation would be really bad. And so things like, things that make up bone, for example, genes that are important for that, if there's a mutation there and that's bad, mutation rates are going to be really low because either you're not going to see those rates because they were removed from the population because they died, or again, these are just, there's a lot of pressure to keep these the same. And so it really depends. So there is genomic variation in mutation rates, even between the other type of genome. Mammals tend to have our mitochondrial DNA, which also can mutate at different rates. So the answer to your question is yes, it does vary, and it varies across species. It varies across different regions within your genome, and it really depends on, again, how important those genes are and how likely they are to change. And because mutations can often be mistakes. So in neutral areas, they are going to often mutate much faster than what we call protein coding regions, or things that actually have a function.
Excellent. Excellent. See, Glenn, Liz knew. Okay. Let's see where I'm at here. Nancy's got the next one. How much does your research rely on previously discovered phylogenetic relationships to determine what each gene means if the species genome hasn't been sequenced yet?
Again, depends on the species. So there are techniques that you can use to find areas. Sometimes we call the most common method right now is to discover SNPs, or single nucleotide polymorphisms, which essentially is, if you look at two DNA sequences, the nucleotide is different between those two sequences. So that's a polymorphism at that region. And so there are many different techniques to do that. Some require genomes, some don't. Having a genome is always more helpful because it's much easier to match where your region is if you know what the regions are, but it's not always necessary. But for phylogenetic relationships, again, that can vary based on your question. So if you're doing species level questions, so you're trying to find cryptic species, for example, it does definitely help to have some sort of knowledge about previous phylogenetic relationships, but it isn't, again, necessary. It really depends on what sort of questions you have. If you're looking at functional genes, and you want to assign a function to that gene that you're trying to sequence, that's where genomes are probably going to be the most important, because genes are so complex in how they interact with each other. And so if you have a genome with your species, that's what's called annotated, or you have a closely related species. So for example, there are many genes sequenced in species that are domestic or are of economic importance. So if I were working with something that's closely related to a fruit fly or a cow, that would be much greater chance of finding a functional gene versus if I'm doing something that is super distantly related, like a badger, where they are so different from any other thing, it would be really difficult to use a genome for them to discover functional genes. So again, really depends on your question, but it is always helpful to know those phylogenetic relationships and have a really good solid genome for sure.
All right. The last few questions that I've got here are specifically about gorillas or badgers. So we might be able to take these pretty quickly. We'll see. In regards to your gorilla research, who or what organization sponsored your work on that, considering it was such a large international effort?
Yeah. So there was the Dian Fossey Fund, which is named after the famous gorilla scientist Dian Fossey. There was also, but I was personally funded under the International Gorilla Conservation Program, which is a organization within Uganda and across the gorilla range. So those two organizations were primarily my funding source. I did have also funds from the University of California, Davis, where I was doing my work through my postdoctoral fellowship that I had with them. So those were the primary funding agencies, but also because Max Planck, which is a university/research institute from Germany, was also located there. So they provided funds for sampling gorillas and things of that nature as well. So there are a lot of different organizations. World Wildlife Fund is also involved. Basically all your major groups that come together to fund this research. It is largely private donations. So certain celebrities have donated to these different groups. Ellen DeGeneres is a really prominent donor to the Diane Fossey Fund. So there's a lot of different ways that this work gets done within that area.
All right. And it looks like the next two that I have here are about badgers. Hank and Denise want to know, how far underground do badgers live?
I mean, they can get 30 feet down. Again, it's hard to know exactly because they dig different types of burrows. So they can have what I started calling central burrows, which is the one where they actually sleep in, they raise young in, they have different chambers to it. So those can go pretty deep, 20 feet, 30 feet. So this would be an example of a central burrow where all the babies are. And then there's burrows that they dig where they're actually hunting. So they're not the fastest animal because they're very squat. So what they do is they will actually dig after their prey. And those can be very deep versus not deep at all, depending on how fast they're able to get that rodent or other thing, but mostly rodents. So really it can be pretty deep and they can have multiple chambers. They actually have an area where they go to the bathroom underground. So that's why I often couldn't get fecal samples, because they have a toilet. But they have a sleeping chamber. They'll often have, different areas, and they can build several of these large burrows. So I found at least 2 or 3 with that. One of the females in those pictures there, where she clearly had dug these very, very large burrows, and they have multiple entrances and things like that. So it can be really complex. Yeah, there's 2 little babies there.
Yeah. And the last one that I've got here, Emelsa writes, interesting talk. Oh, thanks. Great job.
Thank you.
And asked, do badgers compete with other mammals in North America?
They probably do. They're actually more famous for their cooperation with other animals. They're actually known out west to cooperate with coyotes, where they'll actually work together and hunt. But I think probably their biggest competitors are going to be things that are also burrowing, so like foxes and skunks, bears. But they are very well known for their personality. And some of the videos I had during my citizen science I had badgers attacking bears, skunks, opossums, foxes, dogs, driving them off and winning. So they're pretty good competitors, and they're really unique in how they catch prey. But certainly if there's any other medium-sized carnivore or birds of prey sometimes, where they're going to compete with those same prey items. But in terms of burrow systems, nothing really is going to compete with them for their burrows. They often leave their burrows behind, and they're used by other species. So they are important for other species. But in terms of competition, there isn't probably a ton in terms of, as long as there's large species, large amounts of rodents for them to eat. And again, they are not picky. So if one prey species becomes less abundant, they will switch to something else. And so they are probably a main competitor with other mesocarnivores, but at the same time, most mesocarnivores don't behave like they do. So it's hard to know how intense that competition is. But I mean, it is really, the coyote stuff out west, where, the coyotes will actually sit up top and the badgers will dig them out, and then they'll each get an equal contribution. And their hunting is better when they're together versus alone. So it does seem that they have taken advantage of being more cooperative instead of competitive in these areas where coyotes are, at least where it's been recorded out west.
You said that, and that reminded me of this super popular internet clip of the coyote and the badger walking down the drain pipe together.
Yep.
Do you know what I'm talking about?
Yep. So I
I
commented
was
on
trying
that.
to get it pulled up.
Yeah, so that
That's
that was
amazing.
in California at the time. And it was in California, and so everybody was really excited about it, and because that's really what brought people. They were like, the coyote's clearly trying to kill the badger, and I was like, no, they're probably not. They're probably hunting together. And they tend to just sort of have this, they'll have this temporary relationship. They'll get both food out of it, and they just kind of go their separate ways. They don't seem to be at all aggressive with each other, because badgers are pretty well defended against a coyote. They have very loose skin around their neck. So if you try to grab one, it'll just be an amorphous, angry mustelid, because they just have this really thick skin. So yeah, this is my favorite clip of all time. And that's an adult badger. And that's a playbow.
How about
so
that?
they head off together.
Okay.
Yeah. I believe that was, I can't
Next
remember
time
exactly
you're on
where in
Science
California,
Tonight,
but
let's talk about this.
yeah. Oh yeah, once that came out, I was all over Facebook being very excited about, or Twitter and things like that. So people were asking like, is it actually playing with it? I was like, it could be. But yeah, that was really fun when that video came out.
Oh, that's amazing. Well, Liz, thanks for being on the show. This is a great way to go out.
It was a pleasure. Thanks for questions, everybody. That was really great.
Definitely. Thanks, everybody, for tuning in. Everybody, drop clapping hand emojis into the chat for Dr. Kierepka to say thanks. This was a great show. I learned so much. I'm looking forward to actually getting to work more closely with you in the future at the museum.
I am too. Yeah. No, hopefully
I guess I'll
it'll
see you at
be
the staff meeting tomorrow.
Yes. Over Zoom.
Over Zoom. That's it. All right. Well, I'm going to go ahead and close the show down now.
Right.
Liz, thank you so much. Everybody, thanks for tuning in. I'll remind everybody, next Thursday night is a very special presentation. They're all great, but next Thursday is Earth Day. And who better to celebrate Earth Day with than with a museum that has an Earth like on the outside of it. So we're the best place. Come hang out with us. Next week, I'll be talking with Dr. Margaret Klein-Salomon. Dr. Klein-Salomon is a clinical psychologist who's written a number of books about the climate crisis and new and innovative ways to deal with it. So if you're thinking about climate change a lot and you want to learn something new about how we can tackle it, specifically the role of emotions in handling the climate crisis, you want to be here next Thursday. Check out naturalsciences.org in order to get the link, or hey, just subscribe to the museum's YouTube channel right here. Of course, you can follow the museum on social media. We're at Natural Sciences on Facebook, Twitter, and Instagram. Great stuff there all day, every day. And I shouldn't forget, Liz, is there a way for people to follow you and your work?
Sure. I'm most active on Twitter, where it's L Kierepka, so my L with my last name. And I'm also, I have a website, and I'm active on multiple Facebook groups, but I would think Twitter would probably be the easiest. But also, I have my North Carolina state email as well as my Gmail, and more than happy to contact me personally over my two email addresses as well. I'm always excited to talk to people about potential opportunities, as well as just about the different work I'm doing. So thanks again, everybody, for coming to this. It was a lot of fun.
Absolutely. Hey, everybody. Take care. Stay safe. Keep your community safe. We'll see you next time. Good night, everybody.